This is my second lecture on the topic enzymes, and in this lecture I will cover structure and composition of enzymes and various hypotheses for enzyme substrate complex formation. I will also tell you about active site and allosteric site. Now in my first lecture I told you that all the enzymes are made up of protein with the exception of ribozymes which have the catalytic power in the RNA part. The protein enzymes on the basis of chemical nature are of two types. Simple enzymes which are made up of only proteins, and example are urease, lysozyme, pepsin, trypsin, etc. Some enzymes are made up of protein part and a non-protein part, and these are known as holoenzyme or conjugate enzyme. The proteinaceous part of the holoenzyme is known as apoenzyme, and the non-protein part may be a prosthetic group, a cofactor or a coenzyme. Now what is the difference between prosthetic group, cofactor and coenzyme? According to International Union of Pure and Applied Chemistry, cofactors may be organic molecules, then known as coenzymes, or may be metal ions, which are required by the enzyme for its activity and are loosely attached to the enzyme protein and can be separated from the enzyme protein by dialysis. If these are attached tightly to the enzyme protein, then these are called prosthetic group. Cofactor binds with the associated protein or apoenzyme. Apoenzyme is in itself functionally inactive, but when it binds with the cofactor, it forms the active enzyme, the holoenzyme. These are the few examples of some metal ions acting as cofactors for the following enzymes. Magnesium ion is a cofactor for hexokinase. Molybdenum is a cofactor for dinitrogenase, and zinc acts as cofactor for carbonic anhydrase. These are the examples of coenzymes. Coenzymes are organic in nature. They act as transient carriers of specific atoms or functional groups. For example, biocytin is a carrier of CO2. Coenzyme A is a carrier of acyl groups. Flavin adenine dinucleotide or FAD is electron carrier. Nicotinamide adenine dinucleotide or NAD is hydride ion carrier. Pyridoxal phosphate is amino group carrier, and thymine pyrophosphate or TPP is an aldehyde group carrier. Now we come to enzyme substrate complex formation. The formation or existence of enzyme substrate complex was first proposed in 1880 by Charles Adolph Woods. Michaelis and Menton studied the kinetic behavior of enzyme catalyzed reaction on the basis of ES complex formation. Any enzymatic reaction is written in the following order: E plus S forming ES complex or enzyme substrate complex. Then at the active site, substrate is transformed into product, and finally product is released from the active site of the enzyme. This diagram shows an enzymatic reaction in which the substrate binds at the active site, is transformed into product, and the products are released from the active site. There are various models to explain enzyme substrate complex formation. The first one is lock and key model, also known as template model. It was given by Emil Fischer in 1898. According to Emil Fischer, the union between enzyme and substrate takes place at the active site in a manner in which key fits a lock. Substrate is similar to key that fits at the active site, the hole for the key in the lock. This hypothesis is also known as concept of intermolecular fit. According to Emil Fischer's model, active site is presumed to be rigid and pre-shaped to fit the substrate. Modern view of enzymatic catalysis was given by Polanyi, Haldane and Linus Pauling, and according to them, In order to catalyze reaction, enzyme must be complementary to the reaction transition state. It should be more selective to the transition state than to the substrate. And they also presumed that the transition state analogues may be very effective inhibitors for those enzymes. The next model is induced fit model given by Daniel Koshland in 1958. According to Koshland, the contact of substrate at the enzyme's active site induces some configurational or geometrical changes in the active site of the enzyme molecule, and ultimately the enzyme molds itself to the shape of the substrate molecule. Koshland model has gained experimental support for many enzymes such as hexokinase, phosphoglucomutase, etc. This diagram shows the induced fit hypothesis in the case of hexokinase. Hexokinase enzyme has a U-shaped structure. When D-glucose approaches the active site of the hexokinase, the ends of the active site pinch towards each other in conformational change, and this change is induced by binding of D-glucose. Now, what are the active or catalytic sites? Active or catalytic sites are grooves or pockets in the enzyme molecule where the substrate molecule fits. The surface of active site is lined with amino acid residues with their substituent groups. The binding of the substrate in the active site catalyzes its chemical transformation, and then the product is formed. The specificity of the active site is determined by arrangement of amino acid within the active site and the structure of substrate. The interaction of substrate in the active site is by non-covalent and transient forces such as hydrogen bonds, van der Waals interactions, hydrophobic interactions, and electrostatic force interactions. This diagram shows that substrate binds at the groove or pocket in the enzyme molecule. This pocket or groove is known as active site. Active site has particular sequence of amino acids with substituent groups which help in weak bonding such as hydrogen bonds or electrostatic interactions, with the help of which substrate is held at the active site. Now, what are the regulatory or allosteric sites? So, the regulatory enzymes have another site for modulators or effectors. They have active site for the substrate molecule and other site known as allosteric site for modulator or effector. When the modulator binds at the allosteric site, it changes the physical configuration of the enzyme molecule. Thereby, it can increase the rate of reaction or decrease the rate of reaction. So, modulator may be inhibitory or stimulatory. In most of the cases, a stimulator is often the substrate. Those regulatory enzymes in which substrate and modulator are identical, then these allosteric enzymes are known as homotropic. And when the modulator has a structure that is different from substrate, then the enzyme is known as heterotropic. In the case of allosteric inhibitors, generally these are end products of the metabolic pathway, and this end product acts as an inhibitor for the first enzyme of the reaction. This kind of enzyme inhibition is known as feedback inhibition or end product inhibition or retroinhibition. The effect of allosteric modulators is reversible. When these are removed or withdrawn, the enzyme resumes its original activity. This diagram shows the effect of allosteric activator and allosteric inhibitor. If the allosteric activator binds at the allosteric site, the active site has a correct conformation for the substrate and rate of reaction is increased. When allosteric inhibitor binds at the allosteric site, then the active site has incorrect conformation for substrate, substrate is not able to fit in the active site and rate of reaction is decreased. I hope you understood. Thank you so much and wait for my third lecture.